like genomics, transcriptomics, proteomics, and metabolomics. Having all this information from both your pathogen, whether be it fungi, bacteria, or virus, and also the host will help us providing a metabolic network modeling of these pathosystems to the plants. Uh, is 50% recombination was observed for unlinked gene? Is this fixed? Actually, it's like 50% is the maximum number of recombinants or the recombination events that we could see in a population. So above that, we call it as an unlinked gene, and below that, we call it to be linked. But again, it like based on the crop and cross level, we might even restrict 30% as the maximum number of recombinants. But by theory, we can have a maximum of 50% recombinants and 50% parental combinations. What is the best mapping strategy for small labs? Well, it all depends upon the budget and the crop or the species
Yeah, I
understanding.
will come and choose. Let me choose. Yes, I now take their voice.
Uh, Excuse me, can Mustafa, could you please mute your system?
Yes, yes, I muted.
So what's the best mapping strategy for small labs? Well, it all depends upon the budget and the species of the crop and what sequence availabilities do we have. Do we have the whole genome sequencing or some ESTs available? If it is SSRs, they're easy to use in small labs, they're gel-based, and they're established in many species already. All we need is some basic genome information or cross-species SSRs to start with in case we don't have any genome sequence available. On the other hand, SNPs, we have Illumina chips, GBS, and caps and decaps markers, and of course, the resequencing of the entire thing, which are a bit costly. But caps and decaps, we need, with all these SNP based approaches, we can have thousands of markers, well, based on, depends upon the parental differences. They're easy to evaluate because most of the times, if it is a chip, you could get like 50,000 or 500,000 based on soybean or human or whatever chip we are using. We can get data for 50,000 to 100,000 markers in a single Excel sheet in a day or two. So it's an easy way to get that, but it's a bit costly. Whereas GBS needs bit of bioinformatic analysis, it's comparable to the chip sequencing cost. Whereas caps and decaps, they're based on restriction enzymes. We should try out different enzyme combinations to get things. One of my initial project was purely based on caps and decaps. It took us approximately one and a half a year for two of us to use EST sequences, design caps and decaps primers, check the parents, then validate them in the population, and to get a linkage map. My first paper was purely based on caps and decaps. But compared to other things, it takes a bit of time, but at the same time, comparatively cheaper too. And most of the cases, the chips, GBS, or caps, all of these need genome sequences, be it be ESTs or whole genome sequences. If you have specific crop in mind, or if you're not sure about the resources, you can send us an email and me and Divya will try to address that particular species in mind. And some of you have been asking about the softwares. The GitHub link we provided yesterday, workshop requirements, should have all the links that we use. And even we have posted the installation instruction links in that. And all these software don't work in phones. Most of them are catered for Windows systems because that's the common system that we use most of the time. Some of the software, I mean, there are many alternatives to these softwares that we use that could be used in Macs or Linux. And with the data size increase, we might need to look for alternatives. One of you have been asking what's the branch of genomics. Basically, it's a branch of molecular biology where we look for structure, function, evolution, and mapping of genomics. But let's pause for a minute. Uh, First, let's look what a genome is. Genome is nothing but an organism's complete set of DNA is called its genome. And as you know, each human cell contains all the 3 billion DNA base pairs. And we have around 27,000 genes from these 3 billion DNA base pairs, and they constitute approximately 1% of the genome. The other 99% are different regulatory elements, different transcription elements, and many other things. So we have 2%, approximately 1% coding among the genome, and it's the entire DNA. And when we call it genome, we generally refer to the whole genome sequence or the DNA sequence. We call, in general, we call it as a genome, but technically it's the complete set of DNA. And when we talk about genomics, we have to talk about other omics approaches that we have in general. So as you know, every cell has DNA and the study of entire DNA. So here we're not talking about a single gene, study of all the 27,000 genes in, say, humans. We call it as genomics. And there we can have mutations, epigenetic modifications. When we're talking about epigenetic modifications, we call it as epigenomics. And then you have this transcription, which converts DNA to mRNA just at a few restricted sites. And that study of that particular mRNA is called as transcriptomics. So in simple sense, let's assume every cell has the same 27,000 genes in human being. But if you look at each one of them has a different function. So if you consider deeply, the idea is not every gene is functional in every cell, and at the same time, the same gene is not functional in the same way in every cell. I mean to say, for example, a single gene might function different in your liver versus kidney versus heart or eye or anything. So the study of these gene functions, expression patterns constitutes the transcriptomics. And then you have the proteomics, which is the study of proteins, or your mRNA gets converted to proteins by translation, and proteomics is the study of these proteins. And metabolomics is your metabolites, which are the post-transcriptional modifications of your proteins or broken down proteins that could directly affect the phenotype. So these are the different omics. Omics in general refers to study of, so whether we're studying the gene or genome or gene or transcript or the protein varies, and these are the different omics approaches. You might hear phenomics, many other approaches too. Basically, it all, phenomics refers to study of the phenotype. And Divya would answer a couple of questions people have been asking for. Divya, you want to take over for a minute?
Yeah. So the first question that came to me was, does double crossover change orientation of foreign gene in the vector during recombination? So the thing is, like, we should know what exactly a double crossover would be. So normally it is just like less than 2% in the whole 2% of the like, you know, gametes that you expect. Only 1% of the gametes you expect, and only 2% of the products that you expect in a diploid cell. So when there is so you can see like your different types of products, like out, which are the products of meiosis. So this is like, if you see uh, the the whole flow chart actually. So there is ABC, which is you can say, which is from a maternal strand, or ABC, and then a small ABC is from a paternal strand. And when they undergo meiosis 1 and meiosis 2, and then you expect these kind of products, then you expect a single crossover product and a double crossover product. And the double crossovers are a rarity, because occurrence of a second crossover adjacent to the first interval is a rarity across the chromosomes. So so this is a parental gamete where there is no literally there is no crossover. And then you can see a single crossover here between AB, and you can expect to see like you can see the products here. So only the here, when there is a cross between AB and AB, only the A is shifted, only the A gene is shifted, and when there is a cross between BC and the BC, only the only the B's are like only the C's are like shifted actually here, and only the left and the right are shifted when there is a single crossover. But when it comes to the double crossover, only the middle one is shifted actually. So A, B, C are, maybe I would say, they are the three genes that are on the same order. And then you can expect, if there is a double crossover happening between the two, only actually the middle is shifted actually. I mean, sorry, only the middle key is constant actually. Only the A and the C are shifted because there is a double crossover happening between the two. So, and yesterday as well, I answered the same for the question, that the middle remains the same, whereas the left and the right are shifted during a double crossover. So, in a circular plasmid, they can be in different orientation. They can be either CAB or ABC as such. But the gene order, the order of the genes are only predicted from linkage, like understanding the distance or locating the position of these genes. So we calculate the genetic distance, and only then the orientations are like set right. So imagine there is a X gene coming into these ABC, and then definitely the products will change, you know. And the orientation of one of the genes can, so if there is a gene that comes in the middle of in the middle position of B, for example, if the X comes in between B and C, and then definitely the orientation of B and C will change, for example, if it is introduced in the middle of two genes. And or if the X gene comes in between A and B, definitely the orientation of the foreign gene X may not be changed, but only the A and B will be changed. So yesterday I answered the same question, whether the orientation of whether the orientation of this foreign gene will be changed or not. But first thing is, you have to understand what a double crossover is. So the orientation of the middle gene will not be changed, only the left and the right are changed, because a crossover happens between two genes adjacent to each other. So in case of a single crossover, so only one of them is shifted, only the left part is shifted here, and whereas the left side of the gene, which is the A gene, but that is shifted, and whereas here the right is shifted actually. So I mean, if there is an introduction of a foreign gene between these, then the placement, again, the crossover products will change accordingly. So that is the idea. I hope it is right. I'm not wrong about it. So, and then the other question was, what are all the Mendel's law of inheritance? So this, I just pulled out the same slides I explained yesterday. So the first law is the law of dominance. So when you have a tall plant and a short plant, you cross between the two, and the first generation of plants will all be tall. So that will explain the dominance. And when you self-fertilize it, when you go on to the F2 generation, or the next generation further, they segregate accordingly out of the during meiosis, and then you get the following products, and three of them look tall phenotypically, that is visibly they look tall, and one of them is short. So that one of them is short because they inherited the recessive gene, and the visibly you can see the three tall because they all inherited the dominant character. But genotypically, if you look at it, like when you investigate using a DCAPS marker or SSR marker, you can distinguish between the heterozygous individuals from the homozygous individuals, where you can see the ratio of 1:2:1. So the second one is the law of segregation. So when you have like two copies of the genes, only one of them will enter as gametes in the first generation, and then in the second generation they will be and then they will self pollinate, and then the offsprings will be in the combination of will show a ratio of 3:1, like I earlier showed you. So only one of the two copies of every gene, whether it is tall, short, or yellow, green, that enters each gamete Their gametes are the egg or the sperm cells coming from their parents, male and the female parent. So during the fertilization, the gametes unite to produce F1. The F1s are heterozygotes with one gene of Y and Y. And then 50% and two types of, if there are like, if the F1 undergoes fertilization, two types of gametes are formed actually, with each gene having 50% of them. And then you can see all the possible combinations here in the F2. So the next one is the law of independent assortment. So if there are two or more, if there are two or more different genes, and there are alleles of two or more different genes for each pair of traits, say yellow and round or yellow and wrinkled, they actually sort out independently into the gametes of the other pairs. So the alleles of one particular gene received by the gamete will not influence the allele of the another one. So here you can see for a dihybrid cross, like when you have two different genes and only one of them enters into the F1, into the gametic phase. And you can see the segregation as the 9:3:3:1. So this is a visible ratio, which you see in the population. So this is all about the three laws of inheritance. You can always go back to it later, to the slides, which should be available in the YouTube. I hope it answers the question, but if you have any doubt, please let me know.
Thanks, Divya. I hope we have answered most of the questions. If anything else is pending, we'll try to address them through email or maybe in the coming lectures if we feel they're relevant. So with that, we'll just go with the next part of our workshop, where we'll be talking about linkage maps and build a demo of it, building a linkage map. Before I go with that, I just want to mention all our data has been posted on the GitHub site. So this is the introduction to the workshop where you should have all the lectures delivered by Divya yesterday. And say this workshop requirements, this should contain all the software and the download links. And we will be posting our presentations and the question and answer sessions immediately after the workshop. On the other hand, we have posted the linkage mapping raw data Excel file that will be used for today's demo purpose. So the idea is once you complete the demo, you could redo it, try it, and if you have issues, always send us a question and we'll try to address first thing in the morning, tomorrow morning. So with that, let me start a few basic concepts of linkage mapping. I know Divya has covered it. I'll just go with the practical aspects of it. So first thing that comes into our mind is what are the uses of linkage maps or what are the applications of linkage maps? Because most of us who have been into genetics from a while know that linkage maps we have been hearing about these linkage maps from years. As far as I remember, the first linkage map was in 1936 or so, but from the early 1980s, we have different different things, RFLPs, rapids, and many linkage maps constructed for many species. So the question is like, do we still have relevance in today's advanced science? The answer is yes. Because when you talk about advanced science today, most of our scientific work is based on whole genome assembly, and linkage mapping is essential to build a genome assembly. Uh, and other things that have worked or that have been still working include map-based cloning, and for studying the genome organization, structure, and evolution, which in simple terms we can call it as synteny. And the most of all, we have QTL analysis, where we will be identifying genes responsible for traits of interest, which will be our demo or workshop tomorrow, and estimation of gene effects of important economic traits, meaning like a particular gene, how much variation does it explain? So something like a particular gene that is responsible for plant height, that particular gene explains 20% of the variation or 40% of the variation. That's a part of QTL analysis too. And end of all, the complete QTL analysis will be able to use marker assisted selection, which I'll talk in a minute too. So other than QTL analysis, I will try to explain what all other three stand for in simple terms. The first thing is genome assembly. So as I mentioned, genome is nothing but the entire DNA of an organism that is present in a single cell. So to get the assembly, we need to sequence it. And we have no approach that could sequence everything or every chromosome in a single run. So for that, we basically fragment our DNA based on enzyme cutting or sonification or other methods, and we sequence them. Initially, until 5-6 years back, the maximum sequence length we could get is only 250 base pairs. But nowadays, the read lengths with advanced technologies include up to a few kilo base pairs. Recently, the PacBio 2 and BioNano sequencing machines could sequence few kilo base pairs at a time. So end of the day, we will have different reads, as you could see here, that overlap with each other. So for example, if I want to assemble a particular, say, cotton genome, I take DNA from the cotton leaf, sequence them at least 100 times more than the genome size. Say, for example, the genome is 1 GB. So the entire sequence I get is approximately 100 GB. And then I assemble into context based on the overlap sequence, because as you know, our DNA sequence is nothing but ATGC nucleotides only. It is that order that stays constant for that particular plant. And when we assemble based on the overlap, we'll be able to get longer sequences called as contigs. And these contigs are approximately a few kilobase pairs. But that doesn't make up a scaffold or a chromosome. So meaning the next part of contigs, we make up scaffold which are a few hundreds of kilobase pairs or a few megabase pairs. The idea is these contigs, you might have only a little bit of overlap like here. You might have bit of overlap, and we have other approaches like mate pair sequencing, back-end sequencing, yak-end sequencing, so that we can order these contigs into scaffolds. Well, backs, yaks, and all other has been in the past. Nowadays, we have high-C sequencing, CCS sequencing, hi-fi sequencing, and many other advanced methods that could make our scaffolding a lot easier. Earlier cases, when we assemble into contigs, we used to place n about 5,000 ends between certain number of contigs if we can't sequence it. As an example, the initial Arabidopsis sequence that was released in 2000 or 2001, it was only 85% or so complete. It has 15% ends, meaning we don't know the sequence there. So over time, they were resequenced again such that we got the entire sequence. So we have these scaffolds, and these scaffolds are further assembled into chromosomes. For that, we use linkage mapping, either in the scaffolding stage or in the chromosome stage. So as an example, let's take this particular scaffold. Here we have about 10 contigs making up the scaffold. How do we know the order of these contigs? Let's assume we have a genetic map and marker 1 or a particular marker is here, and then we have second marker here, and we have third marker here. So this is the order of the sequence. Well, if we have only one marker, that becomes hard. Let's assume we have 2, 3 here, and we have 3, 4, 5 here. So this is purely based on the linkage map. In case we don't have the linkage map, you don't have the whole genome assembly. All you have is thousands of contigs. As an example, the common bean genome sequence, it has 11 chromosomes. and a few hundred scaffolds and a few thousand contexts. So getting from context to scaffolds and scaffolds to chromosome, we need a linkage map. If you want more thing, I have provided a reference that gives a good explanation of all these things. The second thing, one of the major application was map-based cloning. This is basically Justin Ferris, one of my professors at NDSU. He worked with TSN gene for uh, a particular pathogen in wheat. So the idea is, based on initial mapping, he found a region on chromosome 5B that is responsible for disease resistance based on QTL analysis. But this is a few centimorgans in length and it has many genes or many markers within. And we can only do a linkage map or QTL mapping only if it is polymorphic in the parents. But what if it is not polymorphic? So it becomes harder. So we have other approaches. We use other populations such that we can harvest or we can dissect that, use that polymorphism between other things to narrow down the gene. So for example, in this particular thing, here we are talking about 5 megabase pair of the genome, which might include a few about 100 or 200 genes, since it is wheat. So out of those 200 genes, how do I narrow down to a single gene? So what they did was using various markers, these are all marker names within that particular species, and synteny or comparative mapping with brachypodium, they were able to, D is the brachypodium genome that was already sequenced and wheat was not sequenced then. So using these marker sequences, they were able to get certain positions on brachypodium. If you could see, the first stage was such a small thing and your 100 kb is here, this one, but here your 100 kilobase pair is this one, and this one is only 600 kilobase pair. It reduced from 3 to 4 MB to some 600 kilobase pairs. And further, using other approaches, they were able to identify the TSN1 gene that is solely responsible for this trait differentiation or the susceptibility of that particular thing. And this is the particular gene with all the domains in it. And this is not a simple work. This starting from chromosome 5B to identifying the TS11 gene took approximately 3 to 4 PhD students work trying to find map it using different populations. As you know, developing a population or developing a generation of a population takes 1 year. So when you cross two parents, you get F1, which is not used for linkage mapping. 1 year, then you have F2, 1 more year. So just 2 years, just getting the basic F2 population. And if you want to go with a RIL or a back cross, it's 3 or 4 more years. So this is a continuous work for about 10 years. They did starting doing multiple linkage maps throughout to reach this position. So now we can easily say TSN1 gene, and if it has homology with other species genes, we can always say, well, this is associated with disease resistance to this particular pathogen. The other advantage of this linkage maps is marker-assisted selection. When we say marker-assisted selection, here, this on the left, you have the simple pedigree method. On the right, you have the marker-assisted selection. Basically, the advantage of marker-assisted selection is you could screen just a few plants. At the same time, you could reduce the time. Here is an example that shows reduced screening. Here, if you could see, once you screen for the phenotype, the F2 population, you plant all the plants and you select only few of them to advance further. Whereas with marker-assisted selection, we avoid the phenotypic screening part, which takes a lot of resources, both in money and manual labor. We use a marker-assisted selection in the lab, which approximately takes a week or two based on the resources. Whereas your phenotypic screening, you have to grow an entire season. Let's say you're considering plant height. Until you grow the entire plant, you can't screen for it. So it's 1 year of time. But with marker-assisted selection, we're only talking about 2 weeks or maybe a month. So that's saving time. At the same time, only desirable F3, if you consider here we have been planting, say, some 100 plants. Here we need to only advance, say, 20 or 30 plants. That way, we are reducing the cost associated with advancing the generation. So that's one major advantage. Second thing is, in many generations, we will be using marker assisted selection to reduce the number of plants that could be advanced. So as far as I remember, previously when I was studying my undergrad, it used to be 10 to 15 years for a rice hybrid to develop. But using marker assisted selection, I would say we are close to 7 to 10 years. We were able to reduce at least 3-4 years. Well, it again depends on how much can we reduce based on the trait, diversity between the parents, many other resources that are available. And one other thing, linkage mapping, use of linkage mapping is genome evolution. On the right, I just have an image of a blue tit, which is a blue tit, and which is CCA. And then you have orthologous loci from chicken and zebrafish. So these are the linkage maps. So if we use the same markers across them, you could see that the order is conserved in this particular thing. Whereas here, you could see there is a particular change or change in orientation of the markers. So this helps us understand the evolution of the genomes. So basically here we are using the same markers across multiple genomes. We could identify translocations, duplications. As an example, if you have a polyploid, in my case, I work with soybean, which even though diploid has 2 copies of the genome. So when I map it with the bean that I work with, I found each marker mapping to 2 different locations on soybean. So that way we were able to determine soybean has a duplication compared to bean. Or the other way to say is that duplication is only specific to soybean lineage, not for common bean and soybean together. So that is how we study the genome evolution, or it has been studied a lot previously. So the other major part is QTL analysis. So I would cover from tomorrow. I just want to go with the basics. So basically you have some genotyping, some marker data available, and using this marker data, we will develop a linkage map as you see in this picture. And this is just a graphical representation where the recombination events could be, which I will show you tomorrow too. And this is the phenotyping, or where you collect the trait details, whether be it disease resistance or agronomic characters or anything. And we combine all these to get a QTL map to identify a smaller region on the genome that contains, say, a couple of genes or some 10-15 genes that is responsible for trait variation. So this is QTL analysis is about. So with these applications, even in today's advanced science, be it chromosome assembly or assembly of the whole genome, we need to do linkage mapping. So for that, there are basically 3 steps. One is development of the mapping population. Second is genotyping the mapping population. And third is map construction. I know Divya has covered a bit yesterday, so I don't want to go much details, but a few other things that were not covered yesterday. Mapping populations, basically when you select the parents, they should have contrasting for trait of interest. For example, if I want to study plant height, one of the parents should be a taller plant, one of the parents should be a shorter plant, not both the tall plants or not both the medium sized plants or something. So basically they should have contrasting traits. If it is disease resistance, one is highly resistant, the other is highly susceptible. And then we develop a segregation population. These are few of the crosses that we have been using in diploids from a long while, be it F2s or back crosses, double haploids, recombination in bread lines. And as you can see, a recombination in bread lines takes approximately 7 to 10 generations to achieve high homozygosity to map. So we're talking about 10 years of development. Whereas F2s and double haploids, we can get it in a couple of years. Again, it depends upon the crop. If you're working with a tree like mango, it's not in years, it's tens of years that we are talking about. So the advance from the last 5 years, in addition to those crosses, we have been using magic populations. These magic populations basically are 4 ways and 8 ways. In 4-way cross, basically you have 4 parents. First you cross A by B, then C by D, and then you cross among themselves A, B, C, D, B, C. So you get all these combinations and you get a high recombination. When you use RILs or F2s, we are only talking about a few recombination events. Meaning, if you have a population of 100, and if you're talking about a real population, your final number of recombinants could be just a few thousand or something. But so we'll get 1,000 centimorgan map roughly. But if you have magic populations, we're talking about 4,000, 5,000 recombinations over just 3 to 4 generations. Because you have higher diversity that's being included in each generation, or you have higher recombination spots or higher chances of recombination to happen. Whereas in 8-way cross, it's again cross of these two where you end up having all the A, B, C, D, E, F, G, H, all the segments in a particular set of chromosomes. These have been really relevant. I mean, if we could develop with proper resources, we can get a highly dense genetic map. But we cannot use the regular mapping softwares. These magic populations need a different set of softwares because the way we analyze the recombination or the ratios are totally different. Since this is complex, we're not going to cover in our workshop. We'll just concentrate on the diploids since this workshop was mostly aimed for understanding the basics. So the next question comes is, how about polyploids? We have been talking about diploids. Well, polyploids, when we talk about polyploids, most of the species that we have are polyploids, be it canola, grapes, switchgrass, wheat, tobacco, banana, strawberry, sugarcane, potato, and many other crops. These are just at the top of my head that I wrote down. So they have basically two complete sets of chromosomes, tetraploids or multiple sets of chromosomes. For example, strawberry is an octoploid, and I don't know how many of you know sugarcane. Sugarcane has no fixed ploidy. Like for example, a bean is diploid, potato is tetraploid, strawberry is octoploid, sugarcane can vary from 7x to 21x. And each plant is different. So in these polyploids, we have auto and yellow polyploids. Auto polyploids generally arise within a species and they are homozygous at every locus in the genome, meaning a particular locus is all the same, A, for auto polyploids. Whereas yellow polyploids arise due to hybridization of 2 distinct species and they have varying degrees of heterozygosity. For example, an yellow tetraploid could be AABB. So it all depends upon the heterozygosity, depends upon the divergence of the parental genomes. So the general question that comes into mind is why these polyploids? Actually, during the adaptation process, there were multiple phases in the evolution of angiosperms where most of the species became extinct, particularly at the KT boundary around 60 million years ago. It was polyploids that survived and many other diploid species were wiped out. So, and at the same time, they have higher hybrid vigor and better adaptation ability. And at the same time, they have gene redundancy, meaning since you have multiple copies of a gene, maybe not all of them are functional in multiple copies, but you have a backup gene if some gene fails due to a particular mutation or particular other factors. So with this, polyploids have been a part of angiosperms or part of our plant system a lot. So for this highly heterozygous crops, developing F2s and RILs is a bit hard job. So for this, we can even use F1 populations, which is kind of new if you're only working with diploid populations. which we generally don't use the F1 populations. Here, your parents carry several alleles and the segregation ratio varies in your F1 progeny itself. So just like your F2 reals, you have 1:1 or 3:1 ratios. Here in F1 generation itself, you have these ratios. So this is an example for tetraploids. On the left, you can see the different parental combinations possible, and you have disomic and tetrasomic. These are the ratios, 5:1, 35:1, or what, all these things. So the question comes is what's disomic, what's tetrasomic? For now, let's go with simply. Disomic is for allotetraploids, tetrasomic is for autotetraploids. If you want to know, this is a very good paper explaining all the principles behind all those ratios. And the second system, once we have these populations, whether be it RILs or back crosses or F2s or magic populations, we need a set of markers. One question yesterday was what are dominant and co-dominant? Basically, dominant markers, you have only two allelic states, presence, absence. That's it. And you don't have a heterozygous condition that you can detect on it. Whereas with a co-dominant marker, like for example, SNP or any other thing, or SSRs, you could see parent 1, parent 2, which are homozygous, and you have a heterozygous condition too. In terms of SNP, parent, let's assume parent A has allele T 100%, and parent 2 has C allele 100%. Heterozygous condition is something where you have both T and A at that particular position. And for the marker systems, olden days we have been using many other marker systems, but nowadays SSRs for bit small labs and SNPs that are being used most of the time. When I call about SNPs, we have Illumina chips developed for human soybeans and most widely used crops. You have Illumina chips with 50,000 markers, 30,000 markers, even 500,000 markers. If you look at wheat, soybean, you will see most of the papers based on Illumina chips because you can get 50,000 and in wheat, it's 90,000 markers you can get at once. So rather than using other approaches, it's faster, easier, and you all you get is a simple text file or an Excel file with all the alleles called. So all you need to send is your DNA and you can get the output in a week or so. I mean, on the machine, it hardly takes a day. And then you have the GPS, which is low-level sequencing, and you have next-generation sequencing, where we resequence a lot. And other than the SNPs, we have indels. Basically refers to insertion deletions. So in one parent, let's assume a particular gene is 3,000 base pairs. The same gene in the other parent could be 2,950 base pairs. That 50 base pairs is missing in the second parent. We can use that as a criteria to identify, or we can use that as a marker system by itself. There are many indel markers that we use. Even sequence-based, we can have one base pair indels to all the way a few hundreds kbs of indels. But hundreds of kbs of indels are very rare. So based on the system, we are using a gel-based system. We need at least 20 to 30 base pair minimum for visual differentiation. But if it is sequence-based, we will be using even one base pair or two base pairs or so. And the other one is K-mers. This is a new system of markers that our lab, our group have been using it from the last 4 or 5 years. I'll talk in a minute. And once we get these marker systems, not every marker is useful, and we have to remove the markers that don't provide useful explanation. So for example, if it is RILs, we look whether it's close to 1:1 ratio. And if it is F2, is it close to 1:2:1? So based on the cross and other things, we will be looking for different segregating ratios. K-mers. So basically what K-mers are, short fragment of sequences. This is an example where you have, ABC, these are 3 base pair K-mers. You could see there are multiple K-mers possible just from this sequence. So if you take a 80 base pair K-mer, it could be present in one parent and absent in the other parent. We use that as a criteria So we have all the sequence reads, then we extract k-mers from all the sequence reads, and we create a k-mer table, presence and absence, 0, 1, more like a dominant marker. So this needs a lot of resources, basically a lot of sequencing and a lot of bioinformatic analysis. So basically, k-mer is nothing but a short segment of DNA or a short fragment of sequences. In general, we use 20 base pair, 30 base pair, 80 base pair, depends upon the species and many other factors in the genome. So coming back to polyploids, the markers in, for example, earlier I mentioned SNPs, we have homozygous and heterozygous conditions. And also, these are only 3 states, whereas in polyploids, we have multiple states. They have a different naming system. So when you nuliplex are where you have both homozygous parents. Simplex is you have the simplex, triplex, duplex, double simplex. These are the different marker systems that could be in polyploids. And recently, as I mentioned in sugarcane, it's 7x to 21x. And no one developed a linkage map so far in the last 20, 30 years we know of. It's really, I mean, it's not a good resolution. There are very few markers. So we have been using k-mer sequencing and mostly simplex markers and triplex markers to identify it, to get the linkage map and the assembly of sugarcane going. So when you have this is an example of autotetraploids, and you have nulliplex, simplex, duplex, triplex, and quadruplex. So these are the different segregation ratios. So this is all in just like an F1, 1:1, 1:4:1, and all other combinations. And the software for this polyploid linkage mapping are mostly Polymap, Tetraploids, SNP Map, NetGivas. And here this Burke et al. 2008, this has a very good description in case you're working with polyploids. So other common terms that we hear for when we talk about linkage maps, one of them is map saturation. Basically, let's assume we have a region of interest that's about 5 centimorgans. So theoretically, it might contain 200 genes. We need to narrow the interval to find the real gene, like gene cloning, for example. So if our population does not permit use of more markers, because for regular mapping, linkage mapping purposes, we need the parents to be polymorphic for that marker. As you know, most of our plants or even humans, we are 99% identical. So it's just a few markers that could be available and easy to use. So for that map saturation, we use 2 other kinds of populations. One is called as near isogenic lines. As you could see in the right, you have your BC2s. You continuously backcross with one of the parent until around for 6 to 7 generations until you get this nils or near isogenic lanes. Basically, what happens is, with continuous backcrossing, we're just introgressing only that particular gene. The entire other part of the genome is the same except that particular LD block or gene, whatever you call it as, that is only different. As an example, Williams is a cultivar in soybean. You have 2 other cultivars, Williams 79 and Williams 82. There you have only those phytophthora genes that vary, but rest of the genome, the rest of 59,000 genes or whatever, they're 100% similar. And nils have homogeneous genetic background except in the location of the gene. That's what I have been talking about. But it takes a few generations to develop this. With this, we will be able to narrow down the QTL region. The other one is bulk segregant analysis. As you know, if you have parentals which are generally with contrasting, as an example, we have a high strain and a low strain. You have an F1 that's heterozygous. We take the high trait ones and the low trait ones, leave the middle ones. So for example, I have an F1 with or an F2 with 100 sample size. 10 of them are extremely tall, 10 of them are extremely short. and the other 80 are in between. So I remove those 80 samples out, pool those or combine those 10 DNAs together in each of these pool, and we use that bulk segregant analysis, or we use those bulks or pools to understand the introgressed region or region of interest. As an example, one of my work included a finding candidate white mold tolerance genes in common bean. We know the QTL was present on chromosome 8, but the initial um, QTL was a few megabase pairs. So we took the pools from nils, from nils, and we sequenced them based on SNP frequency distributions. We were able to identify a few regions, and we were able to narrow it down to few genes. The other concept we hear in general is map merging. So in simple terms, let's assume I have a real population of A by B, and I have around 1,000 markers in the linkage map. So let's assume the genome is 1 gigabase pair, or 1 million base pairs, to be simple. So we're talking about 1 centimorgans equivalent to, or 1,000, since we have 1,000 centimorgans map equivalent to 1 million, we have about a few thousand base pairs for each centimorgan. And I could not map further to narrow down any of the regions. Some other lab has some BCF2 population for some C versus D parental lines, and they have some 500 other markers. So similarly, we have other labs or other groups working with other parental combinations, other marker systems. All we need is a few common set of markers so that we can merge all of them together. As an example, in this particular example, your A has 3 markers, B has 4. So you can merge A and B. Since you have 2 common markers, you could merge them and make into a 5 marker system. And then you can merge with C to make an 8 marker system. So when we were trying to build a human genome in the last few years, if we have limited resources for developing a linkage map, because it takes years to develop a linkage map, because it takes time to develop the populations. So if we have multiple resources available, this is one thing we would have been using it. And some of the softwares include AP merge, LP merge, AS map, that's an R package, and merge map. Merge map is something I have been using it a while. So the idea is to saturate the map with multiple markers which were derived from multiple populations. The same thing, for example, if I have to develop this particular map with 8 markers, it might take me to use these markers again into these populations. So we're talking about a bit of time and bit of resources. So this is a particular example, one example in Kajan or PGNP. See where you have different 3 populations, and combining all the markers, we were able to get a decent map out. That includes most markers and better placed positions. So when we talk, now let's go to the main practical aspects of it. The first part is, once we get our data, we should be cleaning for the data. So first thing, parents should be polymorphic for each of the marker we do. For example, if parent 1 contains A and parent B contains A allele, now that's we have to remove them. And the second thing, to study recombination or do linkage mapping is, parent 1 should always have the A allele and parent 2 should always have the B allele. I'll show you an example. I'll show you the practical thing of it. Next thing is, if we have any duplicate marker things, the software will detect it, or maybe we screened the 2 markers because first time it was not good. We screened the marker again and we forgot to remove the data set out. We have to make sure we remove those particular rows that have duplicate marker names. Then we have to remove markers that contain missing data. And then we have to check for the segregation ratios using a chi-square test. 1:1, 1:2, 2:1, or whatever ratio we are looking for based on the population. And When using the mapping softwares, we have to make sure the parental sequences or the parental alleles are not included in the final file. This is an example this is the format for the software. I said Carthagene/MapMaker because MapMaker is the first um, software developed for linkage mapping. If I remember correctly, in 1985 or 1987, this software was constructed and still is used by many labs. It's all command-based. It's a good one, works on a Linux system. But my intention is to teach the basics, so I have been using GUIs. And there are many other softwares, Qtl, Cartographer or R packages, many other things that use command lines to build a linkage map. But as I said, I'm only looking for GUI softwares for training purpose. Well, anyways, the first line of your raw data file should have a line called as data space type space some value. This particular value is the cross. Array self, F3 is F2 intercross or F2 backcross, F3 selfing population. Array is nothing but recombinant inbred population. So we write that. And the second line contains three numbers. The first value here, 46, defines the number of progeny or your population size. The second one includes the genetic loci or number of markers. And the 3 indicates the number of quantitative traits in the data set. Since we are using only for linkage mapping, that's a 0. Or else you could include multiple columns with the phenotypic data too and include that value. I mean, only single value at a time. In addition, for the same line, you could have other symbols. Most of our data, we format the parent one as A, parent 2 as P, and missing as dash. In case you are using a different format, for example, X, Y, or some dash, you could just specify it here, nothing wrong. But this has been the base or this has been the default for most of us. After these first two header lines, then you have for each locus or each marker, you have a star symbol starting with, then you have a marker name, then you have multiple spaces or tab and followed by your genotypic data. As you could see, my parent 1 is A, parent 2 is B, and dashes are your missing data. And this is a real population I'm showing you as an example. That is the reason it does not have a heterozygote. For example, if I'm working with F2s, then I'll be having AB dash H, B, A, H, whatever. I mean, it depends. You can have a H alphabet also in the input file. And here are a few Carthagene useful commands. I don't want to talk about this, but there are many other commands, but these are a few commands that we might be, or some of them we will be using them today, but you might need to use others for your particular project or something. The most important thing to know is like the help. If you type in that, you get all the commands. If you want details, parameters of all those commands, just type your command name. For example, here dsload, dsload dash capital H will provide more help on that particular command. And most of these commands work in default mode. So you can use this particular file to make any modifications to those. But you have GUI based options to modify the things too. So first of all, most common things is manipulating data sets. You need to load a data set for analysis. You can summarize it. You can merge multiple data sets. And once you get the data set, the first thing is we identify groups. And these are multiple things to modify or manipulate loci, meaning markers. It prints them. You can select a subset, you can remove them, you can merge multiple markers if there are many of them, many other things. Based on the application, we will be having all of them. And there are different algorithms available in this particular software. We'll be using one or two. Others are not that good, or it depends upon the purpose. To get a best one, we'll be using one or two of them. That's it. And once you get the maps, we need to verify by flipping. So there are couple of commands to do that: flips and polish. And heap is nothing but what all the analysis that has been done in Carthagene software, everything is saved temporarily in RAM and we call it as a heap. So once we can manipulate heap or print the entire heap or a part of it based on the commands. For example, best print, it just prints the best map from the heap and leaves all others out. But if I do that heap print T, it prints all the 100 maps or 10 maps, how many ever maps it has, it prints all of them. Don't worry about this command lines. This is just an idea. If you want to do advanced things, definitely you need these commands. But for the basic versions or your basic map, linkage map, you don't need any of this. But it's always good to know what else can be done with the software. And you can manipulate the maps. You have a few graphical commands and a few miscellaneous commands. So this is one slide is really important if you really want to do linkage mapping in your experiments. The first thing is, don't worry about this slide. I will show you practically. I just want to list a few things. So in the near future, you can just take a printout of this and just use in the same order. First thing is we load the file. We get the information to make sure we had it right. Then we use marker merge, an optional thing. So what happens is if you have co-segregating markers, which means there is no crossover between those two markers, so the data is 100% correlated, or in other way, your data is the same in both those markers. We call them as co-segregating markers. If you have too many of them, it's a good idea to merge them. Basically means you can it includes one and removes the other one. So the idea is, let's assume you have 10,000 markers and a single group or a single chromosome has 1,000 markers. Estimating likelihood out of 1,000 markers might take a little bit of time. But if you remove those co-segregating or common markers, which we use marker merge, say let's assume we have 100 co-segregating markers. So we're only working with 900 markers at that point of time. So we're reducing the computational, we are reducing the performance necessary. And we have options like loci config, where we can configure different things. And we have loci identify groups to identify groups. Groups are nothing but, in the end of the time, they're nothing but equivalent to chromosomes. For example, Arabidopsis has 5 chromosomes. But our linkage groups could be even 20 or 100 of them. It depends upon the marker density we have. For example, if I have 10,000 markers, there is a high chance I get with 5 linkage groups, each corresponding to a chromosome. But in reality or over the years, based on limited linkage information or limited recombination between the parents, you might have multiple linkage groups. So not necessarily we call them as chromosomes. So we will be identifying groups, and for each linkage group that has more than 3 markers, we do all the steps. Basically, we select that group and we see are there any duplicates. If so, we can merge them at the first step or this step. It all depends upon your data size, data table size. And then we use command called as build FW, meaning build framework map, and a detail to get the details. I'll show you in a minute. And we have other algorithms like AnyLink, genetic, taboo, and other things which we could use, and details of that could be visualized. And finally, we use flips and polish to confirm the order or to estimate the least best likelihood or the least likelihood. And then we use these particular commands to get the final map. Or again, don't worry, we may or may not be using these commands, because once we print the detail, it prints all the maps and the last map is the best one. But if you're confused, you don't want to look at all the 10 or 15 maps it produces, we can just use these commands to get the final order. And then we copy this into a file. So I have included all the Excel steps that we use for data cleaning and all the Carthagene steps as a screenshots, which I will be debugging it in a minute. So basically the data is already uploaded in the GitHub page. So we have parents, a marker name, parental sequences, and some samples. We will be first of all finding if they're polymorphic. I'll show you the comments, don't worry. I did this to show you that in case you want to repeat further. And these are parents that have parent A is not necessarily A in all of them, and parent B is not necessarily A of them. This has been modified, so you'll see this. I need to change this particular slide. And then we'll be checking for marker duplicate markers. We estimate the counts of different alleles, missing alleles, estimate the chi-square value so that we can filter out. As I said, if you don't filter, you might have a dump of 10,000 markers. In reality, it might be only 2,000 markers that could be useful. We do a chi-square test and remove markers that don't meet the ratio. For example, you have 100 sample size and you have 95 individuals saying parent A and 5 individuals saying parent B. In a real population, that's completely irrelevant because it should be close to 1:1. Not necessarily the same, but it at least should be like 55-45, not 95:5, which could be due to the many errors that are possible during our screening of the markers. And how to concatenate them in text patch such that we get a final input file. And this is the Carthagene main window. As you could see, if you type in help, you should get all the commands that are available in this particular program. And the load button loads a biological data set. We select the file. And this particular thing shows that the file has been loaded and what it is, how many samples it has. Again, I'll show you the demo of all these things. And we have data info to look at the information on what it is. And then you have groups, basically splitting the markers into linkage groups. So the basic idea is, as I mentioned, you cannot have more than 50% recombination between markers. If they're greater than 50%, you're talking about different linkage groups, and there are different options you can set here to change those things to get these groups. In this particular Arabidopsis example, I ended with 9 groups, out of which 3 of them don't have only 1 or 2 markers, so they're basically outliers. So we have 6 linkage groups left over to work with. Then we select a group one at a time, and using distance 2PD, we get a table like this. Basically, it's a matrix of all by all distances and a detail. And this is one I used a nice map D. Basically, this builds a linkage map based on the distances itself. And this is what the map is. You have 2 different functions, distance Haldane, distance Kosambi. We will be relying mostly on the Kosambi function. And the one we'll be using a framework map. When we talk about that, when I show you the site, I will explain why we use a framework map. And when we hit that, it runs all of the things and you could see the likelihoods, the thresholds and everything. And this is when you go with search config option, you can change the different algorithm options. So this is for the build FW, frontier, annealing, greedy. These are the different algorithms. You can change the configuration parameters for any of these. So the best map is the one which is last in heap, or you can use these 2 commands to get the best map. So with that, I'll stop my lecture and I'll start a demo. As I said, we need an Excel, or this is the WPS Office, a open source one, which is alternative to Office. In case you need a copy of it, you can easily download. So this is a mapping population data I generated or someone else generated. Some of this is not real data. I manipulated a few markers and things. just to, so not necessarily this is a real data set. So basically in the first column, I have all the markers. Second column is parent one, third column is parent two, and I have a population of 101 samples.
Hello, sir.
Yeah.
There are some questions before we.
Okay.
So I think it's quite simple. So one person want to know like whether these markers are homozygous or heterozygous from the files.
I didn't get you. Can you be clear?
So I think it was a file before that, and this person is asking whether these markers are homozygous or heterozygous.
Okay. Okay. These are all homozygous. Thanks for asking. I was about to say that. This is a real population that we are working with today. In real population, you don't expect heterozygosity, you expect 95% or more homozygosity. So all you see is A and B are missing data. But in case, just as an example, if you have heterozygotes like an F2, it will be a H here.
Okay, and there was another short question as well on whether how do you deal with any segregation distortion in this software? Whether you
We
can
remove
use the
those
software.
markers. We remove those markers. Basically, as I said, we cannot get an accurate 1:1 ratio. You would expect a certain number to change. As an example, if you have a sample size of 100, you expect 1:1 in real. So it's 50:50. Say 45:55 or 55:45 is a reasonable ratio, but not 95:5. But I cannot use the numbers. So statistically, we will be using a chi-square test and use a p-value of 0.01 or 0.05 and remove those markers that don't fit that chi-square value.
Okay, there are more questions coming up, but maybe we can deal with it later. Yeah.
Can you please show some heterozygosity in the file and then analyze it? Well, maybe not today. The reasoning is like this is a real population. I want to make it simple. It's in. I will show you where you can use this heterozygosity thing and how could you use that. There's nothing else that could be changed. It's only the H's that are present. You need an extra column to count the number of heterozygotes. I'll explain in a minute.
Okay. Because she uses, so all the markers from animal science experiments are
Yes.
mostly heterozygous. That's possible. Yeah. And is there any criteria for selecting markers?
Well, basically the availability, if you have SSRs, they should have been published or available somewhere in the database. If it is SNPs, either some, I mean, like we should be generating our own CAPs or GBS data. It all depends upon the crop and the availability of resources. There is no criteria. And the good thing is you can use a combination of SSRs, rapids, RFLPs, SNPs, SINDELs, schemers in a single file.
Yeah. Okay, sir.
Okay. And this is an Excel file I shared with all of you. Here we have a marker in the first column, two parents in the column 2 and 3. Then you have about 101 samples. And since this is a real population we're working with, all you have is A, B, and a dash. If you have heterozygous condition, you'll have H's throughout. Because if you say F2, for example, 1:2:1, you'll have half of them H's surrounding. And I just want to show you these markers, other than the markers, just for an idea, I just placed a few SNPs because SNPs are not genotyped as A and B alleles. You know that they're based on ATGC alleles. So I just placed a few as an example to understand how we could change all these things. So coming to the practical part, first and the foremost thing is we should make sure the parents are polymorphic. So for that, just for clarity and brevity, I would highlight both the columns. because those are the two columns we're testing for. I will insert one column on our left, and there is a command called exact in Excel or WPS, and I'm verifying, are these the same? Exact C2 comma D2. False, they're not the same. So I double click here. So it does for all the markers we have. And then I could simply search for true. Is there any true? If it is true, just remove the marker. And luckily, I don't have any markers that are not polymorphic between the parents. That's the first cleaning step we have to do. And now let's look for a second aspect. Before I do that, I always like to create a backup data. So I'll just copy everything and copy into a different sheet. The second part is, let's make sure parent 1 is A always and parent B is B always. So for that, I'll select the entire data set. Sort by P1 and then sort by P2. So it's all AB, AB, it's all good until you find something here where they were screened a bit differently. Maybe a different undergrad or a different student screening it, or whatever could be the reason. There's nothing wrong with that. We do expect all kinds of these issues. So I'm just highlighting so that it's easy for us to visualize. So this is BA that has to be converted to AB, all the alleles. And then you have the SNPs again, which have different ABs. All the B alleles is A, whereas the parent one is CGT. So this is C to A. So I'll highlight this. This is G to A. I highlight so that I know exactly what cells have to be changed. Sorry if I have selected terrible colors. So the first thing is here you should convert B and A exactly opposite to each other. So I select all these samples. Those control shift right. So I select those particular things and using a replace command, first I would replace A with Z or some other thing. I'll just use Z. So A has been converted to Z. Now I will convert B to A and then Z to B. Ta-da, done. This is all ABs. Parent A is again A, B is parent 2 is B all. Now time to change this. First, let me convert this to A to Z again. C to A and Z to I'm sorry, I didn't replace at all. Okay, C to A, replace all. And Z to B, replace all. That completes that part. And then comes the next one. Many a times we work with at least a week to get this data organized, and we spend half a day to get the final linkage map. A with C, G with A, and again Z with B. And the last part, A with C, P with A, Z with B. So now we have everything parent 1 as A and parent 2 as B. If they were flipped, we changed it. If they were in a different format, sometimes if it is an SSR, you might have something like 450 base per length, 350, 450, 350, so on. I mean, it's the length of the fragment. So we need to change all of them too. So we completed the next part of converting the alleles. Now the next part is, do we have any duplicate markers? So for that, I select all the data, sort by marker, and here I use an exact command again, but I would be using this particular cell B2 with B1. Since they are sorted, if they are the same, I should get a true value somewhere here if I have a duplicate marker. Select this, search for false, search for true. We don't have anything, meaning we don't have any duplicate markers. But it is always worth testing for it, because if you don't do that, and once you start your analysis, you get all these crazy error messages, and it takes us a couple of days to figure that out. So far we've checked whether P1 and P2 are polymorphic. Sometimes if one of the parent is missing, for example, you have to remove that marker to remember that. And we verified whether the parent 1 is A always and parent 2 is B always. And we verified if we had any duplicate markers. The next step is, we'll create a few columns and count different alleles. Let's count, say missing, count, A count, B count, expected A, expected B, I square, if you are proficient in say SAS or R or any other softwares, you could get all of these things done. We just want to be simple so that everyone can try to work on it. So for counting this, we use a function called as count if. I don't know how many of you know all these Excel commands. If not, this is a good time to learn. So count if is a function where you select the cells sample1 to sample 101, comma, what is the character we are looking for, dash. Since it's a character, not a numeric value, we keep it in quotes. Just enter It seems there are 7 missing data for this particular marker. The same thing I use here, count if, select the data, and I'm here counting for B. And here count if data A. So this all should sum up to 101. But just in case if you are using F2s or heterozygous conditions, there we count for A, B, and H. We just add one more column to it. But if it is some tetraploids, we might add based on the analysomic simplex or triplex or duplex markers, we might need additional columns. So just double click here with this plus sign instead of this bold plus. If you have this plus, just double click, it calculates throughout. And in general, we are expecting a ratio of 1:1. So for that, I'll use a command such that based on non-missing data, let's do an approximate expected A to B values. For that, I can do a simple average because, for example, I can do this plus this divided by 2. And let's assume since it is 1:1, these both are the same. So this is the expected ratio. This is the observed ratio. Unfortunately, the problem is you end up having decimals. Like for example, this one, we cannot calculate a chi-square value based on decimals. So let's do a floor value, floor or ceiling. Our floor is nothing but it rounds to the bottom number. Ceiling is something where it rounds to the upper number. So I will use a command called as floor. So this is I'm averaging it and rounding it to 1 digit. So we get these values. So these are all whole values and there are no decimals here since I rounded it to the bottom number. So if a 12.5 will be a 12 and a 47.5 will be a 47. So I got the expected values. I got the original values or the present values. So I can calculate a chi-square value. Chi-square is CHI test. Your actual range is this one. And your expected range is of C2 colon. So the chi-square value, if it is greater than 0.05 or 0.01 limit, if it is greater, it means it's reasonably, it's good enough. But if it is less than that, they are significantly different. So I'm just extending the formula to everything, every other marker. So we did all the basic necessary things to clean the markers. And a few things that we can remove are, first of all, let's remove markers that have a missing percentage greater than 50%. So I'll sort by the missing count, largest to smallest. So in this particular marker, out of 101 samples, 89 of them are missing. So it's useless to have it or it doesn't add any other information. So let's limit myself to say 50 markers. So these all markers can be removed. In general, I include a notes column saying that why I removed that. So if necessary, down the road, I might need to resequence or regenotype them. It helps me. So I would write missing greater than 50%. So not equal to wait till here. I'm pasting the same value. And then we remove markers that have a chi-square value less than 0.01. So sort by chi-square value A to Z. Okay. So for example, see here, we have an expected ratio of 50:50, whereas the real data is 1:10. So 1:1 versus 1:10 is far apart. So you have the lowest p value here. So I say chi-square less than 0.01. Control C. Let me have my cutoff at 0.01. So 34 markers approximately. So these, whatever I have notes for, those are the things I need to remove. So I'll sort by notes. So we started with 141 markers. After removing based on chi-square and missing data, we're left with 91 markers. Now, let's copy. We don't need all this information. All we need is this information. Now, let's copy this data into a different worksheet. And we don't need the sample numbers. And we don't want the parental sequences too. So I will just remove them. So we're left with 91 markers and 101 columns or 101 markers. Now let's make this into a format that could be read by the software. So as I mentioned, our first value should be data space type array self. And the second one is 101 markers. The first, 101 sample size, 91 markers, and 0, because I don't have any quantitative traits listed here. So this is close to our input file, but fortunately your marker data should all be combined. You could use a concatenate command here, but you have to select each and every cell, which takes a bit of time. So for that, I will insert one column, select only the marker data. I have been using Control Shift right arrow, Control Shift down arrow to select the row wise or column wise, copy. And just like Notepad, there is a program called as TextPad that I use mostly. There are a few advantages. It accepts regular expressions, which I will talk in a minute. It does block mode where you can select subset block. Okay, I pasted all this data and these are all tab separated. There should not be any separation at all. So for that, I'll go with search, replace, /t. /t represents a tab. /n represents a new line. Those are all regular expressions we use in programming. So I have to have the regular expression switched on. I'm saying a tab with nothing. And if I hit active document, replace all, it combines everything into a single thing. Again, all I did was, I'm undoing this for a minute. All I did was search, replace a tab, which using a regular expression, a tab symbol with nothing, replace all. Select all, Control A, and I'm copying back to my Excel sheet. And here everything will be copied into a single row. Here, one problem, one means we do that is If the sample is starts with a dash, it considers as a negative number or a value and it gives a name thing. So we need to manually remove those equal to symbols. So Control C, I'm copying, sorry. I'll change the format of this. I'll go to menu, format, cells. I'll go with text. Okay. If I go with text, it doesn't accept formulas anymore. So it's easy for me to remove those equal to symbols. See, it has been removed. 2, 3, 4, 5, I think 6 or 7, not many, don't worry.
Sir?
Yeah.
Yeah, I have a question for the sake of audience.
Sure.
So all these 91 markers are distributed along the chromosomes, right?
Yes.
Okay.
I mean, at least that's the assumption when we start linkage mapping. Maybe in the
Yeah.
ending, it all could end up in a single chromosome or just 2. We never know.
Yeah. Okay.
So now I'm selecting these 2 columns. And I'm pasting it into a document. Save. I'm saving this file. So this is the input file we will be using for this. So briefly, during data cleaning step, what all we did was we first checked if both the parents are polymorphic using an exact command. Then we checked for if parent 1 is always A, parent 2 is always B, and if not, we made the necessary modifications. In next step, we counted the alleles, the missing data, and calculated chi-square value based on expected and observed values. And we removed markers that have a chi-square value less than 0.01 and that have a missing count greater than 50% of the population size. So one question that normally people ask is, how big should be the population? The simplest answer is the larger the better. But if you're thinking in terms of publication perspective, for plants, you need at least 100, not less than that. So once we removed markers based on missing data and chi-square value, we just added a header, what type of population it was, and how many samples, markers, and how many quantitative traits are present. Just in case if you have a heterozygote or some markers are screened as small a, small b for various other reasons, you have to specify using this additional characters in the second file. Okay. This completes the data cleaning part. Now let me start the Karthaji. So if it is a Windows system, you can just search for the program, and if your active TCL is properly installed, the 8.4 version, a single click should open that. So this is a GUI, graphic user interface. So with the help of a mouse button, you could get the program running. No need to write any command unless you really want to make changes and do all those things. So you have a data, loci, search, and maps, 4 different options. And these on the top, or these top buttons are different algorithms or different ways of searching for the map. Each one has its own advantages and disadvantages. On the left are mostly commonly used steps in Carthagena or linkage construction. So let's start with help. That's where we get most of the things. Help. These are all the commands that could be used here. If you want to have a particular command, for example, I want to know about LKHN. All you need is LKHN -H. It's already listed here. I'm just showing it as demo thing. So it gives the description on what the parameters are and what they stand for. Moving on from the help to our real data set, the first thing we do is load our data set. So select the path. In my case, it's in OneDrive, desktop, Vignan. Let me hit OK. If you could see here, the equivalent command in case you want to type your own command is dsload and this part of the file. But right now we're using a GUI, not need to worry. If it loads correctly, it should say what the population type is, RSF, how many markers and how many samples are in there, and the path of the file. If there is an error, sometimes it may not display the error. And if it doesn't display this particular message, the program did not read the file. If you hit on data info, it should give kind of the same information. We have data type as RSL, 91 markers, 101 individuals, and this is the file. So once we have loaded the data into the memory, we have an option called as a loci config. For making groups, what are the LOD thresholds and what is the distance threshold? Distance LOD is nothing but a LOD 3 means basically 1 by 1000 error, 0.001 p-value. A LOD of 2 means 0.01 error value. So the higher the better, but to start with, we always start with a 3. But based on the marker system, crop and everything, if you have a LOD of 10, meaning the error rate is 1 by 10 power 10. It's a very, very good map. But to start with, let's do a 3. And the minimum LOD of your final map should also have a LOD value of 3. Minimum LOD to get a publication or to get a decent thing out of it. And the distance threshold, 0.5. 0.5 is nothing but the amount of recombinants possible. As I mentioned earlier, we can only have a maximum of 50% recombinants. So that is that 0.5 value. For example, for the same Arabidopsis, I have 10,000 markers. So I could narrow down these to 30 centimorgans or 0.3 threshold. That way I can get a better map rather than including all the markers possible. But the defaults are good enough for now. So I'll go with OK. Coming back here, loci, identify groups. So basically that's nothing but it's saying these markers can be grouped into 9 different linkage groups. But out of which one linkage group has only 1 marker and 2 others have 1 and 2. So it's basically 6 linkage groups we have to work with. In reality, these 6 could correspond to 5 different chromosomes, 1 for each, and 1 linkage, 1 chromosome being represented by 2 linkage groups. Since the number is too low, it might be 3 linkage groups, meaning the same marker. Combining these linkage groups and chromosomes is based on the sequence of the markers again. But let's not go to the concept of chromosomes. Let's stick with linkage groups. Don't worry if you don't catch up with any of these steps. I will repeat these steps again. So we did groups. Now let's select one large group. Let me start with the 5th group. Before I go that, I always like to copy my stuff so that I can keep track of things that I have been running. So let me start with fifth group. So for that, there are comments. Let's not worry about that. Select a group. And here I just select the fifth group. Okay. So it's saying it's selected marks, it selected all these markers that have been included in linkage group 5. Now, if I go with loci, distance 2 point, basically it's an all by all matrix of the distances. Okay, it's too big to explain. So let me get back one step. Sorry about that. Select the group. Let me take say 9, which is a bit shorter, easier to understand. Loci distance. Okay. If you could see, it gives distance of all by all. For example, if you have 0 in any of these values, meaning they're co-segregating or they're just beside each other or there is no linkage between those two, I mean, there is no crossing over between those two markers. In this particular case, all I see is this M44H and G4715 have a value of 0, meaning they're both co-segregating. If you want, we can merge them using loci, merge two locals and view both names. But right now, I don't want to do that. Rather concentrate on the main other things. So the easiest algorithm or the fastest algorithm is nice MapD based on this 2-point distance. It quickly calculates a map. But to show the advantages of other algorithms, I'm doing this, but in theory, it's not necessary to do this. Oh, it's done. So all I need to hit is detail. Okay. It's a single map because there's nothing else before. And it's approximately 70.6 centimorgans. And the log 10 likelihood is minus 159. This is not necessarily the best map. Many times, if you do this nice map D, your Kosambi distance can be greater than 50 centimorgans, which is not reasonable. So just to, let's go with the main algorithms we work with. Say if I go with build, I start with 3. What happens is whenever it adds markers, the log value might go down to say 2.5 or 2 or even 1. Build command still continues the same way, even the log gets down. For example, we get a decent map, but if the final log of the map is only 2, there is no use of it. And this annealing, taboo, or all other bit advanced, which take a bit of time, but for our smaller data set shouldn't take much time. The best one to get thing is called as a build FW, which is a framework map. Basically, we start with a 3 lot and your final map is also 3 lot. So for that, to start with, I'll go with search, config. Here you have different algorithms where you can configure different the defaults. For example, build FW is something we are using right now. The keep threshold is a lot 3, add threshold is 3, meaning the map should have a lot of 3. And when you add a marker also, it should have a lot 3. Let's talk about flips later on. Build FW, cancel. Okay. Just hit build FW. Boom. It's done. What happens is it starts with 1 mark, 2, 3 markers at a time, keeps on adding, and finally we got some map of it. To get the details, hit the detail. So it's saying this particular linkage map is 50.1 centimorgans with 6 markers with a likelihood of 111. Just bit to be curious, if I did the nice map L, the likelihood is minus 159, whereas this advanced algorithm, the likelihood decreased, which means it's really good. So this is my final map. I'm not sure if this is good enough. So I want to try one algorithm called as annealing. So it did annealing with different combinations and likelihoods. Again, hit the detail button. It's the same 50.1 centimorgans and 111 likelihoods, minus 111. But if you see here, there are 10 maps in memory, or even more, sorry. There are many maps with this any link, and you could see each one has a different distance, different likelihood. But if you see, they are sorted by likelihood, and the last one is always the best. As an example, this is 111 here, 114, 115, so on, and it keeps on increasing. So the first maps are not something we're interested in. It is this last map that we are interested in. If you quickly remember, even with Bill FW, we had the same 50.1 centimorgans and 111. So our framework and annealing, both algorithms are saying the same thing. Just to confirm the order, we do a thing called as flips. We'll work with the defaults. So basically what it changes the order of them and make sure the likelihood does not change much. If it changes, it goes with flips. And here it did not change. I hit the detail again, 50.1 centimorgans and 111, it did not change. And again, I do a polish, again a detail, same thing. So the map initially created by build FW was reasonable. So I'll just copy this, Control C. I mean, you can save the session too, but I'm just copying. I'm a little old school. So what's this linkage group 18? So we work with 9th linkage group. So this is, I only look for Kosambi. Halden is a different function. Anything is fine, but most of our work will be based on Kosambi functions. So we had 6 markers to start with. We had 3, 4, 5, 6, around 14 markers. Out of 14 markers, only 6 markers are included at LOD 3. If I go with the LOD 2, maybe 4 or 5 other markers will be included, but I'm not interested to create a low quality map because anything we do will have repercussions with our downstream QTL analysis or anything else. So we're done with one linkage group. Let's try a different one. Now let's do the 5th one, which is bigger. Coming back here again, loci, select a group, 5th group, okay. We don't want to look at the distances or anything. All we look is to start with is if you want, if you can look at the distances to see if there are any duplicates, but for our demo purpose, I'm not using that anymore. I'll just hit left W. Okay, this is the final order of the markers. It was that fast. To know the details, detail. There are 18 markers that are included at 74.8 centimorgans. The likelihood is minus 203. If you're not sure, again, you can run an annealing. It takes a bit of time because this is a bit longer. Boom, done. Detail, minus 203, 74.8. Maybe a better map, definitely a better map annealing. If it is the same, it would give the same thing. Flips, polish, detail. This is the final map. And here 18 markers have been included and we're done with 2 linkage groups. Now let's select the second linkage group. Again, loci, select a group, the linkage group number, okay. Build FW, annealing, flips, polish, detail. It's that simple. 4 buttons, click, 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 and you're done. We're done with 3. So we're done with 2 now. And left with 6 and 7. Search, sorry, loci, select a group, 6. Okay. Build FW, annealing. Oops, not enough for flips. So, okay. The final marker list has less than 4 markers, so it's not able to do flips. And we did a polish ISO, and it's a detail. Only 4 markers have been included. Loci, select a group. One last one that we'll be doing, that is 7. Okay. The same 4 buttons again, and 4 markers have been included. I think it's the same thing I did again, read it again. It's a different one. And again, as said, the last map is always the best one. So let's suppose these are the 5 linkage groups we are working with. The other ones, I mean, we're only left with one. So let's not go with it. Two things, I mean, how to interpret these results. One thing is this particular GAPB marker is 15.2 centimorgans from this, meaning there are 15.2 recombinants between these 2 markers. If it is 100, exact 100, you will get mostly whole numbers. And 13 centimorgans between this M254A and this M235. But when we do the final map, we need to do the cumulative thing. So to do the cumulative thing, the easiest way, the best part I like here is you can select a block post, double click there, select a block, delete. So that way you can get rid of all the unnecessary things and just keep that for further work. So this is the linkage group 1 with 6 markers that total to a 50.1 centimorgans. And all we have is marker distance between 2 adjacent markers. This is not the cumulative. If you want the cumulative, you need to calculate it. The way we do it is that will be a 0, that will be 15.2, that will be 15.2 plus 13, 28.2. So on, the last marker will be 50. This is our cumulative values that we would be using if you want to develop a map or graph a map out of it. So that is linkage analysis. I know first time users, it might be a bit complex or too much. So I want to quickly repeat it again from the start. At the same time, each and every of this step is being recorded. At the same time, the functions and everything have been in PowerPoint presentations with the formulas. So let me close this. Delete all this. Start again. So if your data, initial data is clean, it's a lot easier for us to work. So let's start with the load. Select the file. So it's loaded. I made sure it's in there. To confirm data info, just to confirm it's loaded. If it is empty, meaning there was a problem in loading the file. Now the first thing I do is identify groups. I have this and copy paste. And then I select one group, and for this making groups, you can use the config window to change the load and the thresholds, distance thresholds. So select a group. Let me take the second group, for example. Okay. Once I select the group, the best algorithm that could be used is a build FW, which has such, if I go with config, build FW, which has a threshold of 3. Based on your requirements, you can change it to 2-on-2 or 5-on-5, 2-3, 5-3, whatever, any combinations. But it's better to maintain a minimum of 3-and-3. Okay, just hit the button to confirm if get the details of this map, and you get 16.6 minus 171. To see if there is a further better map out of it, I use an annealing algorithm. Flips, polish, detail. Boom, this is our final map. Copy paste. Again, for the second thing, loci, select a group. Let me do the last one. Okay. Build FW, annealing, flips, polish, detail. And this is always the last map is the best one. Just copy paste. In case if you're confused with these too many maps out of this, as I mentioned, there is an option called as best print. But if you want to use a command best print, this is the final map. And if you want the details, best print D. So this is the details. It's the same thing, 50.1 centimorgans minus 111, the same thing. So it just prints the final map all by itself, so that you don't need to have the hassle of all these things. And the good part is you could save the session as sorry, I couldn't hear. If someone has a question.
ಕ್ಲಾಸ್ ಹಾಕೊಳ್ಳೋದು ಏನು ಅಂದ್ರೆ ಅವರಿಗೆಲ್ಲ ಅಲರ್ಟ್ ಮಾಡಿ ಕೊಟ್ಬಿಡ್ತೀನಿ. ಏನೋ ರಿಡ್ಯೂಸ್ ಮಾಡಿದಾರಂತೆ ಸೆಕ್ಷನ್ಗಳು ಬಂದ್ರಲ್ಲ, ಈ ಕಂಟ್ರೋಲರ್ ಇರೋದ್ರಿಂದ ನಮ್ಗೆ ಇನ್ನೂ ಈಸಿ ಅಲಾಟ್ಮೆಂಟ್. ಸರ್ ಈಗ ನೋಡಿ, ಈಗ ಫೈನಲ್ ಇಯರ್ಗೆ ನಾವು ಇಷ್ಟು ಜನ ತಗೊಳ್ಳೋದಲ್ಲ.
Can someone pause the person?
ಏನ್ ಮಾಡೋಣ ಈಗ? ಏನಾದ್ರು ಟೈಮ್ ಟೇಬಲ್ ಏನಾದ್ರು ಕಳಿಸ್ರಿ, ಏನಾದ್ರು ಬಂದ್ರೆ ಈಗ ಅವ್ರ ಕ್ಲಾಸ್ಗಳನ್ನ ಹೆಂಗ್ ತೋರ್ಸೋದು? ಆಫ್ಲೈನ್ ಈಗ ಬರೀ ಫೈನಲ್ ಇಯರ್ಗೆ ಅಂತ ಮಾಡಿದ್ದಾರೆ. ನನ್ನ ಸಜೆಷನ್ ಅಲ್ಲ, ಇವ್ರಿಗೂ ಕೊಟ್ರೆ ನಾನೇನೋ ಸೇಫ್ ಅನ್ಸುತ್ತೆ. ಇವ್ರಿಗೆ ಏನ್ ಮಾಡೋಣ? ಗೆಸ್ಟ್ ಫ್ಯಾಕಲ್ಟಿಗೆಲ್ಲ ಎಲ್ರಿಗೂ ಕೊಡಕ್ ಆಗಲ್ಲ.
Bobby ma'am, Abraham sir, can someone pause it?
ನಟ ಸರ್ ನೀವು ಬರ್ರಿ. ಮೀಟಿಂಗ್ ಅಲ್ಲ ಸರ್, ಈಗ ಅಲಾಟ್ಮೆಂಟ್ ಅಷ್ಟೇ ಕ್ಲಾಸ್ಗಳು. ನೋಡಿ ಸರ್, ಎಷ್ಟು ಸೆಕ್ಷನ್ ಕೊಟ್ಟಿ ಅದು ಕುತ್ಕೊಂಡ್ರಿ ಒಂಚೂರು. ಕುತ್ಕೊಂಡು ಟೈಮ್ ಟೇಬಲ್ ಇವೆಲ್ಲ ಎಕ್ಸ್ಪರ್ಟ್ಸ್ ಸ್ವಲ್ಪ ನೋಡಿ. ನೀವೆಲ್ಲ ಕುತ್ಕೊಂಡು ನೋಡಿ 3 ಸೆಕ್ಷನ್ ಏನು ಅಂತ ಹೇಳಿದ್ರು ನಿನ್ನೆ. 6 ಸೆಕ್ಷನ್ ಇದ್ದಿದ್ದನ್ನ 3 ಮಾಡಿದೀವಿ ಅಂದ್ರಪ್ಪ. ಈ TPN ಸರಿ ಒಂದು ಕ್ಲಾಸ್ ಕೊಡಿ, ಒಂದೆರಡು. ಅದೇ ನೋಡ್ಬೇಕು ಸರಿ ಕೊಟ್ಟಿದ್ದಾರೆ.
Sorry about that. So as you could see, it's just 5 or 6 buttons, and it hardly takes, I mean, based on the number of markers, less than a day to build a very good linkage map. There are many other alternatives, whether QTL cartographer is a good one. In R, there are different packages, there are different Unix and different things. It all based on the markers. For one of our Kamer study, we started with approximately 10 million markers, and Carthagene and R are not the softwares that could handle that. So we used our custom Python and awk scripts to filter to, I think, around 50,000 markers that we used finally. I mean, we narrowed it down to nulliplex or simplex markers, and we used only them. So just to repeat, I'll start loci, select a group, 7th group. Okay. Build FW, annealing, or you could skip the annealing too, doesn't matter. Flips, polish, detail. Okay. First question that comes to is, can we represent this map in a graphical format, as someone asked? We can, but that's horrible. So we have other things. Actually, the QTL software that we use really gives us good maps. So we'll be using it there. And again, for graphical representation, you need to convert this Kosambi values into a cumulative value, as I mentioned earlier. And in TextPad, using block mode, or you can use Excel to type manually all those values and markers. So we end up having a final good map. So with that, I'll stop. I will take questions from you if any one of you have it. For sure, since it's the practical part, you'll have a lot of questions. And this is the first workshop that we're doing it virtually, so it's new to me too. So using the same link that we provided yesterday, submit your questions and we'll try to address first thing tomorrow morning.
Participants can unmute themselves and can ask the questions.
Well, someone asked how could there could be so many missing data? Well, this is just an example data set created out by our own, but in reality, you might have, for example, SSRs or something due to DNA loading problems or the marker itself, because not everyone is good at wet lab experiments. And at the same time, the marker it might not be polymorphic or that much representative. So we end up having missing data.
Hello. I just want to ask that. Hello. I just want to ask that if these are the 6 markers that are mostly linked, and you are saying that these are the best markers, so we will use these 6 markers for making the map and ultimately for the QTL detection.
Yes, for QTL detection, basically I have a different data set, because I just want to start this mapping with a small set of markers rather than going with a few 500 or 1000 markers and sitting hours and hours for it to analyze. For QGene or QTL analysis, we will be using a completely different set, which has already been analyzed.
So currently, these markers, just we need to show that these are the best markers out of all the markers we are using, and they have the like they are linked with each other. Do you want to show these type of the things for this lecture?
Sir, could you repeat again, ma'am?
Just saying that we just did this linkage mapping. So in the linkage mapping, just we are saying that if we want to conclude from this one, so we are concluding that out of like these 100 markers, these are the 6 best markers that are showing best linkage with each other.
Yes.
Okay.
So basically we started with 91 markers, out of which we identified 9 groups. We identified 9 different groups, and out of them we had 6 groups more than 3 markers, which might correspond to 5 linkage groups. And for each one of them, some of were not included because of a low lot value. For example, if you're not building a framework map and that is starting point for something, you could decrease the load and have more markers included. As an example, in this particular thing, at a load 3, I had only 3 markers. If we increase the decrease the load to 2 value of 2, then we might have 5 or 6 markers included in this. And not necessarily all the markers we start with end up in linkage groups due to various things.
Fine, sir.
I have a question.
All right, sure.
Hello. Yeah. So my question is regarding, it seems you have been relying on a lot of students to produce data which is being used by the private sector. So how reliable is it, like, using the student to produce data, which is actually going to be relied upon? I mean, do you make replicates, or, I mean, is it reliable enough?
Depends upon the students. And nowadays, I mean,
I
like the
see.
previously, the SSR markers and all those gel-based things are mostly replicated and generally done by PhD students. So they're generally, I would say, 90 to 95% good. But if you're using SNPs or Indels or next generation sequencing, we're talking about 95 to 99% reliability.
Okay, thank you.
So with this, we'll conclude the linkage mapping part. And tomorrow we will talk about QTL mapping. So what if we have a linkage map and some phenotype available for the same real population or F2 population or whatever it is? How do we come to a conclusion, or how do we identify the region on the genome that is related to this, to the trait under study? Could it be disease or plant height or anything else? So for that, download the software QGene, and it needs a Java JDK as a prerequisite. It should work on Mac and Linux systems. And if you have questions, feel free to fill them in the form that we provided yesterday.
So then, sir, can we end the session, sir, for today?
Yes.
Divya ma'am, do you want to share something?
Nothing. Actually, I request everyone again, once again, to access the software simultaneously while Sir is demonstrating. So this will help you to run through the process, with your hand, actually. So although we cannot give like a hands-on workshop, since it's a virtual workshop, it's very difficult to connect you guys personally. So kindly access the software simultaneously while the demo is going on, so that when you encounter some problems, it can be asked. And I guess that would be more efficient in putting your questions as well as giving the right answers. Thank you.
I think I can end of the session today.
Bye, everyone. See you all tomorrow.